If we move inside of the cytoplasmic membrane, what we will find is cytoplasm. Cytoplasm is the gelatinous material that fills the inside of the cell, and this gelatinous material is made up of two parts. We have the liquid part, and then we have all the other stuff that's in the liquid part. The liquid part is what we call cytosol. Cytosol is made primarily of water, and then we have a lot of other substances dissolved in it. That would include maybe some ions, maybe some sugars, some proteins. If we were to label that on this picture, it is the green part. Okay, so this is all of the liquid that's filling the inside of the cell, inside of the cytoplasmic membrane. Now, there's lots of stuff in the cytoplasm besides just the liquid component. So if we talk about some of those things that are actually in there, one of them is the DNA of the prokaryotes. The DNA, it is going to be a double helix. In prokaryotes, it's actually a large circle. Okay, so it's a large circular chromosome, and so if I were to draw it, it's a double helix, so we've got that typical double helix structure. Not the best drawing, obviously, here, but it's this big circle of DNA, and then it's going to be kind of wound and twisted together in this region that we call the nucleoid. So here it is the yellow component inside of this cell. Another thing that we have here in the cytoplasm is going to be inclusions. Inclusions are what we call deposits of various molecules. Deposits of lipids, deposits of starch and glycogen. A lot of food storage molecules will get deposited here. It's not specifically shown in this picture, but we could just draw in little dots to represent the inclusions that would be there. And then another thing that we can have in the cytoplasm of bacterial cells is endospores. Endospores are not always going to be there, but they certainly can be. These are going to be really durable, really resistant structures that can survive a lot of really harsh conditions. And so if there's, it's really dry, they can basically survive and then kind of regenerate somewhat into a new cell when conditions return to something that is going to be favorable. If we were to look just really quickly at how these endospores form, at the top here we can see that first up is we'd have to have the DNA copied. So the genetic information has to be copied, number one, and then the rest of this is basically taking that extra copy of DNA and packaging it up in this really resistant, durable structure. So that's what we see finally at the very bottom down here, is that we've got this little structure. It contains the DNA inside of it, and it's very resistant. And again, these very resistant little spores can survive harsh conditions, and then when conditions are favorable, then they regenerate into a normal bacterial cell. Continuing to talk about the cytoplasm and some of the other things that we find in the cytoplasm, non-membranous organelles are what we have in bacteria. We don't have organelles that will have a membrane on the outside when we're talking about the ones in bacterial cells. Okay, so things that we do have, first off, is ribosomes. Ribosomes are the little blue dots in this picture. They are where the proteins are going to be synthesized, or in other words, this is where the proteins will be made. You can have lots of ribosomes inside the cytoplasm, and that's really what we're seeing in this particular picture because there's lots of little blue spots throughout. Another thing that we would have is the cytoskeleton. The cytoskeleton is this internal scaffolding of fibers, long ropes that you would have throughout the inside. If I was to draw it in this picture, it'd kind of just be like this, extending throughout the inside of the cell. These are made up of different types of proteins, and function-wise, a lot of them are going to be for transport purposes, helping to move things from one side of the cell to the other side of the cell. They also help to provide kind of just an internal framework or internal skeleton, if you will, which is where this name comes from. So that's going to provide some support for the inside of the cell. Let's talk now a little bit about Archaea. Archaea are our second group of prokaryotic organisms. And bacteria and Archaea, they're pretty similar to each other as far as their external structures are concerned. But one big difference with Archaea is that none of them have been conclusively proven to be pathogenic. So disease causing. There are some suspects, but again, none have been conclusively shown to be pathogenic. So if we talk about what we do find around the outside of Archaea cells, they are going to have glycocalyces. So that's gelatinous, sticky structure made of polysaccharides and polypeptides that we find around the outside of the cell. The functions of the glycocalyx is going to be for sticking. So it is going to help them to adhere or stick to other cells, other archaea cells, or to other specific surfaces, which could be non-living surfaces that they're actually growing on. They do also have flagella. Flagella, if you'll remember, are really long fibers, kind of long tails that they can use for movement. So these would stick outside of that cytoplasmic membrane. These do have some structural differences compared to what we would find in bacterial cells. So they're not exactly the same. They provide a slower movement for sure. They're much thinner in diameter, so about half the size of what the bacterial flagella would be like. And the actual components that make them up, amino acids specifically, that is going to be different. So they're made of a different material, and they're going to contain some sugar molecules attached to them. We didn't have sugar molecules attached when we're talking about the ones that you find in bacterial cells. So the bacterial cells, theirs again is made of flagellin. That's a specific protein. And for these, they're going to move using some ATP. In contrast, when we're talking about movement with the bacterial ones, it's a gushing of ions that are flowing out. This is not going to be a gushing or flowing of ions through the membrane. It's the ATP that's actually going to be used to provide the power for this slower moving tail that we have extending from the cell. With these, they're also going to rotate in unison both directions. With the bacterial cells, we had tumbles and runs. That's not the case here. These all are going to work in unison in both directions, so there'll be kind of runs when they're going both directions, no tumbles where everything kind of jolts to a stop. Other external structures, there are fimbriae. Those are those sticky-like, rod-like projections, bristle-like projections that extend from the cell. They're going to help them to stick to each other, stick to specific surfaces. And then a unique structure that they have is the hamus, or hammy for plural. These are tiny prickles, but the unique thing about them is if you look in this picture here, it has like a little fishing hook kind of grapple on the end of it. And so these are really good for grabbing hold and sticking to something. They will help the cell to firmly attach to specific surfaces. If we talk some about the archaea cells and their cytoplasmic membranes, their composition is a little different from what we find in bacteria. Archaea cell walls are going to be made up of specialized proteins or specialized polysaccharides, and that causes the shapes of the cells to be different from what we saw with the bacterial cells. So here you see just some of the variety that we have. We have even some that are more cube-like or box-like, which is the third picture here. And then if we're talking about the actual membranes, the membranes are not going to be made up of phospholipids. These membranes, they don't have phosphate groups in them at all, and they have what we call branched hydrocarbons. So structurally, they're different, and this structural difference, the way that it's linked together, the membranes are linked together, we call it ester linkages, and these are much stronger, more stable linkages when we're talking about extreme environments. So extreme temperatures are going to be okay when we're talking about archaea because of those special linkages. So what we find is that these tend to live in a lot of environments where other organisms can't survive because the membrane of other organisms won't hold up in that particular environment. So these, a lot of them are found in extremely high temperatures, like in hot springs. Some of them are found in really salty environments. Just really extreme environments tends to be where you find your archaea organisms.